Hi. So, in previous session, in previous several session, we have studied about the unique properties of immunity. We have learnt about various immune organ, and in immune organ we have studied thymus, bone marrow, spleen, lymph node, lymphatic system, and mucosal associated lymphoid tissue. Now we will move on to the the cells of immune system, which which is playing a very important role in defense, and the generation of these cells, generation and development of these cells, we call it as a hematopoiesis. So, if you remember my previous session, I have shown you that this hematopoiesis is started immediately after a week, and it is basically taking place in yolk sac, and thereafter it is taking place in fetal liver, and subsequently it is taking place in spleen, and after the birth of the fetus, it is taken over by the bone marrow. Okay. So, this is very important to understand this hematopoiesis, and basically all this hematopoiesis or development of blood cells, which includes both immune and non-immune cell, that is RBC as well. All these cells are the development of all these cells or generation of all these cells are taking place from the one of a very unique cell, which we call it as a hematopoietic stem cell, and hematopoietic stem cell is very much essential for the reconstitution or constitution of these blood cells. Okay. And it is very interesting in adult human, this hematopoietic stem cell is residing in the bone marrow, and there is a one cell in 50,000 cells, in 50,000 bone marrow cells, only one cells are there, and these cells are generally in very less number, and this number keep on changing, depends on the need. Okay. Need means if there is a severe loss of blood, then these stem cell will divide, and they will increase in number, and immediately this cell will replenish the blood cells. Okay. And then again they will reduce in the number, and these cells has some unique marker, and these markers are playing a very important role in isolation. So, after discovery of this unique marker, now we can isolate the hematopoietic stem cell, and this hematopoietic stem cell we can use it in various kind of therapies. Okay. And in fact, we are using this marker in order to isolate the hematopoietic stem cell when we are performing various experiments. Okay. Here just I am giving a note about the mouse, because in whatever immunological studies we are doing, we are mostly doing using the mouse model. So, here I will talk about some of this mouse experiment as well. So, if you irradiate the mice with X-ray with 950 rad, which is a unit for the radiation, then you can deplete all the hematopoietic stem cell, as well as you will deplete all immune cells in the mice. Okay. And if you deplete these cells or if you irradiate this mice, then this mice is highly vulnerable to the infection, and subsequently they will die. Okay. So, we use this this strategy for doing various kind of experiment in order to prove some of our discovery or phenomena. Okay. And if you irradiate these mice with this radiation X-ray, and then if you can infuse these mice with say 10,000 or 1 lakh of these hematopoietic stem cell, please note this stem cells should be derived from the syngeneic mice. So, there is a very big difference between the syngeneic mice and other mice. Okay. Syngeneic mice is those mice which is generated after breeding from brother, sister and all those things. Okay. So they are so their genome is kind of consistent. If you take the group of syngeneic mice, their genome is kind of consistent. Okay. There is no variation. So in that scenario, you if you will take some cells from one mice and then put it in another mice, then there will be no reaction. These cells will be taken up by that recipient mice. Okay. But this is not true for human, right. So we are outbred. Okay. So syngeneic mice are basically, in a simple term, they are inbred mice. Okay. And in case of human, if you take some tissue or cell in general, then and if you transfer it to the another recipient, then this will develop a reaction, and those cells or those tissues may be rejected. Probably you may know that before transplantation we do lot of analysis for tissue matching and all those things. So this in case of syngeneic mice, it is not needed. And if you have a group of mice which is irradiated with X-ray, you can rescue these mice, because after irradiation these mice will not survive, but you can rescue these mice by taking this hematopoietic stem cell from syngeneic mice and transfer it in these mice. So you can rescue. Okay. And this number of cells are very less, it is about 10,000 to 1 lakh, which is approximately 0.01% to 0.1%. Okay. So using this strategy, I will explain you one experiment. For example, you want to investigate a particular cell type that play a crucial role in defense against viruses. Okay. By a lot of experiment, by several things, you are speculating that this particular cell type is playing a very important role in defense against, say, some particular virus. Okay. Let's talk about keep an example of NDV, Newcastle disease virus, okay, which is not pathogenic to the human. Okay. So what we can use this strategy, and for example, this is a group of syngeneic mice, and using this syngeneic mice, the genome will be consistent. Okay. And you can divide this syngeneic mice in several groups. So for example, here I have made it 3 groups, and then in one group you can infect with virus, another 2 group you can irradiate with X-ray, okay, with 950 red. Okay. And from infected population, you can isolate the cell which you have discovered earlier, which you have discovered, you can isolate those cells. There are a variety of techniques by which you can isolate that particular cell if you know the marker. Okay. For example, you can use the magnetic bead system, or you can use the flow cytometer. Okay. Using these techniques, you can isolate those cells, and after isolation you can infuse these cells in one group of radiated mice, and in another group you can introduce the cells or the cell type which you have discovered from uninfected mice. Okay. And you will if after doing all this thing, if you infect this radiated mice, then you will see that the group of mice, the group of irradiated mice which received the cells from infected mice will be protected, and another group will be not protected. So in that way you can show your discovery. You can demonstrate that the cell which you have discovered in by various way, it's fitting well under the physiological condition. That is most important. So whatever experiment you do in vitro, that is not so good. So in that scenario, you have to prove it in your discovery under physiological condition, and in that way you can do you can prove that okay, this cell is playing a very important role against the virus infection. Okay. Now I will talk about some various type of stem cells. So these are totipotent cells. So totipotent cell is basically just derived from the egg, fertilized egg. Okay. So it's basically the cell at early stage immediately after fertilization. So these single cell, first it will become a zygote, and then this will divided into 2 cell stage, 4 cell stage, 8 cell stage. So at that stage, if you isolate the cells, so these cells has a potential to make a whole organism. Probably you might have seen, or you might know that there are mirror image twins. Okay. So by some during embryology, after fertilization, during development, somehow this zygote divided into two parts, and that generates 2 individual, and those individuals are mirror image twins. Okay, they are identical, almost fully identical, right? So those cells we call it as a totipotent cell. Another is pluripotent cell. So these are the most cell type, like a human pluripotent stem cells can give rise all kinds of body cells. Okay, but it cannot make a placenta. Okay. So from this pluripotent cell we can make a various cell type like a bone, liver, and all those things. Probably you might know that these days the doctors suggest to preserve the stem cell immediately after the birth. But I would like to say that it's currently we don't have a technology, or we don't have things which directly used in the clinical setup. Okay, it is all in a discovery phase, or people are standardizing all over the world. Okay. So this pluripotent cell, we are, in case of mouse, we are using very frequently, and we call it as an embryonic stem cell. And we can manipulate this embryonic stem cell, and we can make a variety of transgenic mice. In next slide I will discuss all those things. Another is unipotent cells, and these unipotent cells are very restricted development steps. These unipotent stem cells can make only one kind of cells. So that is why we call it as a unipotent stem cells. Okay. Another is multipotent stem cells. So these multipotent stem cells can make a few cell types which is quite close to each other. Okay, biochemically or in terms of function. Okay. So those cells we call it as a multipotent cells. Okay. Now I will talk about the pluripotent cells, because this is quite commonly used in our research. Okay. And one of the aim of this course is to educate you for research also. Okay. So this pluripotent stem cell is basically very commonly used in mouse experiment, as I have explained to you, and we can generate the transgenic mice. Okay, using this pluripotent stem cell, or in case of mice we call it as a embryonic stem cell. So we can create a variety of transgenic mice, and these are you can create knockout mice. Okay. So knockout mice is I would like to explain that this knockout mice is nothing. It's just you, for example, you discovered some gene, and this gene is playing a very important role in particular phenomena. Okay. So what you do by using molecular biology techniques, you just inactivate this gene, and you can do it very easily. Not so easily, it's quite difficult, but it is possible. You can inactivate the gene UTR, or you can inactivate the exon, and in that way this gene will be inactivated, and we call this kind of mouse as a knockout mice. Okay. Another is knock-in mice. So, knock-in mice is another very interesting reagent to prove some discovery. I will explain these two mice in subsequent slide by showing us very simple experiments. Okay. So, in knock-in mice, what we are doing, we are introducing some gene at appropriate location in the genome. Okay. For example, you want to you might have seen in videos or in research paper, there is some mice which is giving a fluorescent green color. Okay, when you radiate this mice in blue light. Okay. So, those are basically a GFP knock-in mice. So, what we do, we simply put the GFP gene under the control of that promoter. Okay, promoter promoter of that gene which is expressed in the skin cells. Okay. So, in that way we can make a knock-in mice. So, that is the little fancy thing, but I will tell you the importance of knock-in mice in discovering new cell type or variety of new things. Okay. And all those things you can do it under the physiological condition. Physiological condition means in the animal, and once you show it in the animal, then everything is foolproof. Okay. There are different kinds of knockout mice. One, we call it as a conventional knockout mice, and this conventional knockout mice is basically you just inactivate that gene. Okay. So, once you inactivate that gene, then this gene will be inactivated in all cells of that particular animal. Okay. But sometime what happens, if you inactivate that gene, and if this gene may be involved in development somehow in development process, then this will cause the embryonic lethality, means the after fertilization of this egg by sperm, the embryo cannot develop because this gene may be involved in development process. Okay. So, in that scenario we make another kind of mice which we call it as a conditional knockout mice. Okay. So, conditional knockout mice is it's a far more complex, but here I will explain you in very simple way. What you do, you basically inactivate that gene in particular cell type. Okay. For example, there is some pathogen or some antigen is there, and this pathogen and antigen is basically taking playing an important role in macrophages or dendritic cells. Okay. So, when you are creating a conditional knockout mice, you will knock out this gene only in dendritic cells or macrophages, not other cells. Okay. So, this is a very complex technique. In next session I will discuss how to create this conventional or conditional knockout mice. Here I will just explain you how these mice can be used for proving some very important discoveries. Okay. For example, why transgenic mouse is needed. Okay. So, here one so when I say transgenic mice, it could be a knockout or it could be a knock-in. So, first I will just tell the importance of knockout mice. For example, if you have a question or if you want to investigate the role of some signaling molecule by so many in vitro analysis, bioinformatic analysis, you have discovered some molecule which is playing very important role against the virus infection, and now you want to prove it under physiological condition whether it is valid or not. Okay. Because when you perform the in vitro experiment, at that time you miss so many things. Okay. So, in order to prove it under physiological condition in animal, you need to create a knockout mice. Okay. So, for example, this is a wild type mice. Wild type mice, when I say it is not wild, it is not catched from the wild, it is also a syngeneic mice, but they have a they are having a gene, the intact gene. Okay. The intact gene for whatever molecule you have discovered, and this is a knockout mice. Okay. In this mice, you just mutated the gene or you inactivated the gene. The most appropriate term will be inactivated the gene. so that that gene which you have discovered is not functional. Okay. And then you have infected this mice with virus. Okay. So, for example, the gene which you have discovered, and it is playing a very important role in defense against the viruses. Okay. So, what will happen? The wild type mice will survive. Okay. But on another hand, the knockout mice will die and it will not survive, or it will die quite early. Okay. So, in that way you can prove that okay, this gene is playing a very important role in defense against the viruses. Okay. And subsequently when you will do all those things, you need to prove it at various level. So, for example, you will investigate all antiviral factors, you will estimate the antiviral factor after infection using wild type or knockout mice, and then you will see that in wild type mice the antiviral factors are very high, and in case of knockout mice it is almost abolished, or it is present in very low amount. Okay. So, in that way you can prove it. Another experiment which is quite commonly people use is to check the viral load. Okay. So, since the knockout mice does not have an appropriate defense system because of mutation in that gene or non-functional of the gene, so you will see that the viral load will be extremely high in that mice. For example, this mice is basically infecting the liver, okay, or lungs. Okay. So, you will take out the lungs and liver, and you will compare the viral load in using wild type mice, and then you will see that the viral load is extremely high in knockout mice. Okay. So, in that way you can prove it that okay, this molecule is playing a very important role against the virus infection. So, this is one example. Another example which I would like to say about the knock-in mice. For example, you have discovered a cell type which is playing a very important role in against particular virus infection or bacteria infection. Okay. So, how you will prove it? Okay. So, what you will do, you will discover or find out the gene which is exclusively expressed in that particular cell type. Okay. And you will create a GFP knock-in mice. Okay. You will put the GFP molecule in that, and then you will perform this experiment. You will take this the wild type mice. Again, it is a it is having an intact gene and there is no transgenesis, and you will use the knock-in mice, and then you infect with different viruses. Okay. And after that you will isolate that particular cell type. Okay. And then you can show that this in case of this virus infection, in this cell type there will be there is increase in number of the cells. Okay. So, in that way you can say that okay, this mice is playing a very these cells are playing very important role in that particular virus infection. It is a quite huge detail. I am telling you, I am just giving a glimpse of how you can use this transgenic mice in order to answer the questions in immunology. So, in next session I will discuss about the various type of immune cell, but before that I will take up how to create these transgenic mice. I will just give you the glimpse. I cannot teach you in a great detail because it's a huge amount of material. Okay. Thank you.